Good morning, one and all. This is Dr. Ashish Shruntla, affiliated to Department of Basic Sciences, SR University of Warangal. And here is my contact details. If you face any troubles, any difficulties in understanding some part here, feel free to ask any questions. In the session 6, the first part 6A, we'll be extending our ongoing discussion. Last session, we have been discussing about phylogenetic profiling and protein-protein interaction network. And why should we do it? Normally, what happens whenever we are interested in finding out the function of a protein, what the students or researchers do, they do a blast search and keep on checking the function of top ranked sequences. But many times, what happens distant sequences present in the same species may also share the functional details with this sequence. So it's always helpful and always intriguing, interesting part to find out the set of proteins which are working together in the similar pathway, in the same pathway. Okay, so this will help us to understand how these proteins are mutually working and that way you will understand the functional details of a protein in a much deeper way. Understand? So for doing that, phylogenetic profiling make much more sense. Remember that whenever you are doing phylogeny, it's just only building a evolutionary tree. But when you are doing the phylogenetic profiling, we are only connecting the presence or absence of certain genes in different species with respect to different functions. So, in multiple species, if the gene is present, this means there's somehow some connection between the two, which means that they are probably the protein partners or probably involved in the some sort of biochemical association either in the same pathway or the connected pathway. Understand? Remember it properly. All right. So, but now when to use phylogenetic profiling and when to use protein-protein interaction network. Remember that whenever you have less number of genes, okay, less number of genes are there, like 4, 5, like 6 or 8, then you can choose phylogenetic profiling. But you have much more number of genes, say 100, 50, 200, like that. And then you want to come down to functionally crucial protein partners. So the most important strategy is to build the protein-protein interaction network. Then you can narrow down to like few of them to decode their like phylogenetic profiling, to decode them like functional associations further. Okay, that is how people do. All right. To understand that further, here I have given the definition also. That you always remember. Phylogenetic profiles, when you are saying, is to identify the profiles that are mostly related or have similar patterns of presence or absence across different species. It suggests that they may have similar functions, they participate in related biological process, as I have just been telling you. Okay, to predict the function and characterize proteins. Simple. It's only the presence or absence, just to indicate that they may be involved. I'm not saying they must be involved or they will be involved. They may be involved. But protein-protein interaction networks, large number of genes, it provides a comprehensive view of physical associations. Okay, it may be it get the data from experimental data or predicted data or gene altered data. All those things are taken care. Besides that, under what category, what part of the cell, where exactly in the cell, all these things are considered here to decode the functional association between the proteins. So that's very important to integrate all of them, integrate both the data so as to decode the functional details much more better way. Okay, but now you understand that whenever we are doing all these things, we require that our database information highly correct, highly updated. What it means highly updated? This means that whatever the sequence I'm searching, okay, any sequence X, my database, whatever I'm searching, if it is not properly updated or I'm not using the desired set of databases, I'll not have the required data on my table to take the information from. Understand? So people always need to connect the database so as to search the things here and there everywhere. Like if I missed something in the house, okay, miss my file or miss my copy or something important note, I check out everywhere in the table. Reason is I just don't want to leave any chance that it may be present in the house, but I have left it. Okay, keeping something ignored as such. Okay. But these things are constantly growing, and that's the biggest challenge. Protein-protein interactions are very temporary. So many databases, many peer-reviewed journals, or databases derived from the peer-reviewed. Okay, secondary databases. They also have some information which are not in the primary database. So I need to always connect them and then get the derive the information further. Okay. So this always need to like always connect different type of data which are shared by different researchers, following their own like patterns. Like because many scientists use different patterns or nomenclatures. Okay, so when you connect all of them together, we follow a proper unifying strategy, single strategy, just like IUPAC name is there. But in biology, people have been using diverse sort of names. Okay, so that's the major trouble. So this has been the first database, see, BioGrid database. Basically worked out for Saccharomyces cerevisiae, Saccharomyces pombe, Candidal beacons, and some other organisms in humans. See, when you see this database BioGrid, see, this is the part it looks like. BioGrid statistics means how many sequences are deposited there, how many entries are there, when it got updated, not all those things. Here you can put your protein or gene name or the accession ID if you have. This is the result turning up. See, if I search LWR here, I am getting this result. Gene ontology means for 16 different biological processes it was involved. For function, 4 different functional details have been found and like available in the databases. Go component means where exactly in the cell it functions. So I've got 3 different components in the cell where it is found to be functioning. Okay, see, shown here. See, condensed chromosome, outer kinetochore, condensed nuclear chromosome, centromeric region, and nucleus. People have submitted these 3 details there. 67 proteins, 25 publications are there. See, so you can get many different things at one shot. When you go in more detail, see, further down the page, if you scroll it down, you get more details. Like the sequence, here the colored part shows the serine and threonine have undergone the post-translational modifications. It includes the phosphorylation, see, and taking up the information from these papers. So it gives you the information about like which amino acids have got post-translational modifications. Done. I got the list. You won't get it just like that. Moreover, when the information got published, that also you can get it here. See, along with the proper source, proper link. That's the advantage of connected servers when you integrate the things together. See, moreover, it is giving me the complete protein-protein interaction network. How it is there, see. But when you link all these proteins in set of clusters which are much more associated with each other, you find complete genes are associated in 2 clusters. This is cytoscape tool. If you learn it, you will enjoy the things. All this is part. Okay, see? It is showing me two clusters here. All the genes are associated with two set of functional mechanisms associated here, see, and connected to this. See, to take it further, this is the part which I have taken from Briefings in Bioinformatics 2010 paper. You can refer to this article. See, this image shows the interaction network between several different proteins here. Now, when you analyze it further, see, so many different pathways are involved in. This is the advantage of clustering. As I have shown, cytoscape is used in other things. All the cytoscape clustering helps us to associate the proteins with respect to functional clusters. Okay, like if multiple people are present here in the classroom, suppose. Okay, and I say, how many are you? You are the best friends. Everybody won't club in a single group. They will be clubbing in several different functional groups. Wherein much more in much more some connections is there. Exact there it is, just like emotional bonds. Here it is chemical connection or the some sort of biochemical relationships are there, biochemical interactions. Okay, to drive some functions. All right. This is the second database, molecular interaction database. See, for the humans also it has been separately worked out, termed as homomint. Okay. This is how it is worked out. See, if the Mint database finds that one protein in the PubMed or like already deposited like a published research papers, there some protein is shown to be connected to some other protein, forming up a stable complex. Then what is that interaction which is connecting both of them? First thing, and how the thing methodology has been there, which is shown to be connecting this. These few details are properly analyzed and then deposited in the Mint database. All right, Mint database properly keep on reading various articles, keep on tracing through various articles to keep on update the databases. All right, if the protein has got modification, how exactly it has gone, where exactly, all the details. See, moreover, the protein domain, the structural domain, and the taxonomy. See. Now to see here to demonstrate how the Mint database works for a protein. See here, P53 when I'm searching here, very common protein. Okay, so cellular tumor antigen, then tumor suppressor P53, soft protein. This is now it is named as see 393 amino acids, Homo sapiens, giving the functional details, nucleus it is found. This detail we get in the biogrid also. But see, post translational modifications, we get it there also. But do you get it here? GenBank, ProSite, PFM, PDB. How and what thing is connected here? What all sequences have been deposited connected to this information in various data sets. This information was not there in that database. BioGrid did not give us this kind of thing. It only showed us some papers which are published at resource, but nothing related to various sequences. In much more detail, see, it is searching much more. And then it's connecting here various parts, see, all these proteins are connected. Now see, this is the database which I personally like too much in, the string database. Here see, it is much more sophisticated database, which I especially like because see, besides getting the data from the published network, published research articles, it also extracts the database from some predicted information. Say suppose one protein is closely associated to four other proteins, but if a close homologue of that protein I have, what is the chance that will be like closely connected to other proteins? Maybe quite likely they will be connected. Okay, so it just builds the extend the homology between the various proteins to generate their network. Okay, following the network of some proteins in a species, which is very closely related. I hope you understand. If you get the like functional network of some protein in one species, okay, in the other closely related sequences, there also, even if different species, orthologs, you transfer the information, okay, and then work out like these proteins may also be connected there also, okay, just to build up the initial data. Whether or not they are connected, actually, that you need to see it in the experiment or need to do further check. Understand? But it just extends the part entire possible way to get you the required thing on the table. That this may be the right path. Alright? Because whenever you're doing metabolic engineering research, there you need to get entire single information which is currently there so that you build your further research approach. The major part 14,094 organisms, the data is there. 1025 bacteria, 1597 eukaryotes, 472 archaea. 67 million. See, these many proteins, maybe 20, more than 20 billion interactions. Now, these interactions, it also assigns a confidence score. Confidence score means something which is experimentally shown to be there. It assigns high confidence. But something which is just shown to be predicted with the similarity. So if the similarity score is very high, or of course the confidence may be increasing. Okay, confidence should increase. But if the similarity score is only 20%, 30%, similarity score goes down. Okay, so the confidence also goes down. But it may be there. Saying database is not saying it is not there. It may be there. It is not escaping from the chance. Okay. Genomic context, whether the genes are there or not there, high throughput experiment, conserved co-expression. See, means the proteins are always getting expressed and they are conserved also. Means whenever I am expressing this protein, somehow the other protein also gets expressed. Means this protein X is always somehow involved in connection with the protein Y. Then only whenever I'm working on X, Y also comes up. So XY protein complex will certainly be working somewhere. Or the previous knowledge, see? So either something are functional physical connections or something are indirect connections. See? This indirect association includes everything which you can predict. Alright. See? Now see the part which I'm getting here. Results. See functional inference set system level. See? This is how you got. Gene neighborhood, gene fusion experiments. Okay. Like you combine the two genes and then clone it together to see whether they are working together or not. If they work together, their protein products will always be forming a complex co-occurrence. If this gene also occurring, that gene will also occur. See, experimental data, different databases, text mining, homology. Okay. Text mining is from the published literature. See, and from various databases connecting all of them. So by doing homologous search, one can map a known biological pathway in one organism to another one. It's like I have been explaining to all of you. Hence, predict gene functions in the context of biological pathways and networks. You can always connect. Many ways exist to find the putative function of a protein sequence. It is not known, it may be it is putative function. Okay. For more reliable prediction, it is important to combine many different prediction protocols. is used by the string server. I'm using all of them to just to put up a scale of confidence. Right. See, this is the second part. See, now I'm showing you how the confidence score is there. See, besides how this functional details is found out that this protein is connected to this, what is the a proof? This is the proof here. That somewhere it has been shown to be done through text mining, and text mining probability is 0.96, highly credible. See? Normally people use over 0.6, but if you want to check out two divergently connected, very distantly connected proteins, you may even go to zero point 0.4 or zero point 0.5. Now do we'll do the concept check. Okay. Which of the following is a primary objective for protein-protein interaction network analysis? Determining the 3D structure? Of course not. Identifying post-translational modifications? This is not the right answer. Exploring functional relationships and modules means functionally connected modules in a cellular system. Of course, it makes sense. And predicting protein sequence modules. This is not the right answer. Option C is the right answer. What is the main purpose of analyzing protein-protein interaction networks? Identifying the primary sequence of proteins known, determining protein localization within the cell. For this, I can use the gene ontology search. I hope you are understanding properly. The gene ontology search we can use properly. Predicting protein secondary structure. I'm not at all touching all this secondary structure by protein-protein interaction networks. Understanding the organization and dynamics of cellular processes. That may be the right answer. That's the right answer. Okay. So constructing and analyzing network graphs is the common approach used in protein-protein interaction network analysis. Yes, it's right answer. Okay. If so, can it give wrong results? And should the data be experimentally validated? When can it give the wrong results? When you take many different proteins, even if they are connected or not connected, but you put them together, which people normally do also. Okay, so experimental validation, firstly remember that experimental validation is always required. After doing all these networks, you do mutation analysis, you do site director mutagenesis, and work out whether these complexes are actually forming or not forming. First thing. Moreover, do after building up an initial map with many proteins, you try to build up the cluster, just like more functional association proteins, just like I have shown you here. You build up the functional clusters and then discard the others using the confidence scores. Okay, use this confidence score to build the clusters and discard the outliers which are least connected to your proteins. Alright, this is a common approach which is used. See, after learning so much, now we'll be moving onward how to build the complete pathway. Okay, so what are the different models? What is the different strategy to do it? Once that is done, can we find out is it right or wrong, or can we simulate the model to under various conditions, like in the system itself, rather than doing the same checks in the actual cells, where it may be too complicated or too much time taking or involving too much money. All right. Now remember that we are extending our ongoing discussion of protein-protein interaction network to metabolic network, so that I can drive the complete part on a new track, something which you can enjoy for metabolic engineering aspect. Okay. Metabolism is the process involved in the maintenance of life. It is comprised of vast repertoire of enzymatic reactions and the transport processes used to convert thousands of organic compounds into various molecules necessary to support cellular life. These are taken from Schilling et al. 2000. This is the complete reference. Here, like, see the entire pathway how it is worked out. Like, it involves 7 steps altogether. First is the genome annotation. First step is to annotate the genome of the organism of interest. I need to work out. Okay, so I need to identify and annotate the genes, the coding sequences completely. What is the intron portion and exon portion, entire part. Once that is done, then I'll do a database mining. Okay, I'll explore further what are the existing databases and resources present for these sequences. Moreover, once that is done, I'll find out the metabolic reactions which these proteins or enzymes are involved in. Once that is also done, I'll compare the existing databases to see if there is some known metabolic pathways or known some missing information somewhere there. Like I'll tell you, like recently we're finding like Rubisco enzyme or RUBB part is present in E. coli also, bacteria also. So RUBB is plant. RUBB is present in plants for photosynthesis. Okay, which is capturing the light part. So what it's doing in the bacteria? So photosynthetic bacteria is also there. Is it having some connection there? So sounds interesting. Like that, different gaps are found, and there is a need to bridge the gaps. Okay, so through constant research, we always like bridge the prevalent gaps, which are existing gaps in the metabolic networks. This network only allows us to extend the protein networks. Recently, you have seen many different proteins connected to P53. Earlier, it was not there. Okay, but when people are finding that some proteins are connected, now some proteins are itself connected to some other proteins, means they are there for a second layer of interaction networks. So this allows us to extend the existing protein networks. Okay, and then building the cross connections. Like that, complete metabolic engineering is worked out. Alright, it's a very interesting subject. Now see the fourth part here. When you say model assembly, whatever you have done so far, okay, you worked out a computational model. You generate the computational model. This involves building the proper mathematical equations, right? Like writing the complete part that how many genes are involved, how many metabolite molecules are involved, putting up the reactions all together, build up a computational model that under these conditions, these things, these prevalent existing set of features or parameters, like these genes are properly guiding this metabolic network. Okay, once that is also done, you move to the like model validation and refinement. This means that like seeing the existing data set, seeing the existing part, we we check our validate our like computational model for the conditions which you have found in the lab. If the data is fitting or like properly we worked out or we fine tune. Okay, this refinements are iterative process. Okay, so we undergo iterative process to extensively refine the like generated computational model. Okay, once we build all the mathematical equations, then we go for the simulation step. Okay, we analyze and simulate. Once that is also done, then we go for the last part. We use all of them to like check out, like apply on a like biological conditions for some different species. Once we find that some information is wrong somewhere, some things are not performing properly, means initial data set which we used for gene annotation was wrong. Some genes were extra there or some genes were not at all there. Okay, so we extend the network again or delete the like unwanted genes which are not playing any significant like significant role in the simulations. Okay, so we try to connect all the bits and pieces just to get some sense out of it. All right. So why is metabolism required? Metabolism is the like, complete soul mother of the life. Okay, without metabolic reactions, there is no life. See, this is the cell membrane I am showing here. Different molecules come in and go outside. Okay, different reactions take place, and this is happening in each and every cell of the body. So now you can understand in a multicellular organism things are so much complicated. See, I'm showing here amino acids and proteins. See, carbohydrates. See, nucleotides, lipids, coenzymes, minerals. See, so many categories of molecules, and which are woven together. See, like the nature has woven them together to guide some certain functions properly at a proper rate, and no fluctuations or no changes happen. Otherwise, it will lead to like fatal damage. All right. So, it is an essence of life and tremendous importance in medicines. For many diseases, like are unable to control some certain enzymes, certain paths. Okay, some functional networks go wrong, like obesity or diabetes. We are unable to control the glucose concentration in the blood. Okay, the bioengineering aspects are also there. We like a design science for the production of biological products or generation of biofuels. You generate the biofuel at an increased rate, something which normal cells cannot work out. You generate the biofuel molecules like isopropanol, propanol at a much higher quantity. Okay. Now the metabolite, as I told, the key classes. All these classes are written here. See, and the biochemical reaction. See, the process in which one or more substrate molecules are converted. See, now we are going in the details of metabolic network. So many molecules can be combined together. Now see the magical story of connecting the pathways. Alright. See a high isoleucine, valine, leucine, all these biosynthesis. It's showing here so many proteins, various reactions, various pathways. It seems to be like so much complicated. But when I show you the entire part, see, connected in a cellular network. Mind like the mind is completely boggled here, see? But that's how it taken from KEGG database. See? So much complicated. So the protein or the networks, like under various conditions, the things are really complicated. But it's interesting to know about them and extend them as much as possible, so that we can decode the functional nature of various genes and various proteins under various conditions. See, extending the discussion further, if we see a similar network for E. coli, see how many proteins are actually connected together, like such a mind-boggling network. Amongst this, if I just see here, see these proteins I'm showing here with arrows means directed. This is a directed network where ultimate protein, last protein is guiding the towards this one. It is being further connected with this one using this arrow. Some connection is there in this rate. I'm sorry. So this is how the complete biochemical pathway goes on. Which protein guide to what? Which protein is leading to what change? Okay, so some direction is there. But see here, here I am not at all showing anything. Means this is a non-directed or undirected graph, undirected network, where I am just showing the connection here. Alright. So see, with this part, we are now extending our ongoing discussion of extending the how the metabolic network is constructed using the 6 or 7 different steps. So with this, like how to initiate it. See, remember that whenever the different stimulus or different signals or different biomolecules are present within the cell, how to initiate it from, or what should be the initial set. So to understand that, what we normally do is we consider or we prioritize the signals which are coming from outside. Okay, and we consider this a seed set. That's the word of exogenously here. See, something coming from acquired from outside. Okay, so if something is outside acquired, that cannot be synthesized in the cell. So then we will check out the stimulus or the thing which is caused by the outside seed set. Then we'll keep on connecting the proteins or the signals which are like induced due to the seed set. Just like the glucose. Glucose, if it is entering in the like cell, hexokinase is released. Okay, so we understand that, but it is released because of glucose. Okay, likewise. Now see, what are the ways to like quantify the interactions between two proteins. If the two proteins are there, either a sequence or structure, different scoring methods have been developed. I am showing here very like a preliminary method which are actually used and which are very useful and which do not require too much like calculations. First is the phylogenetic closeness. If I'm calling that in the terms of plates, like sequence similarity or average branch length. Sequence similarity will be there between the every two sequences. If n sequences are there, NC2 combination matrix. Like I'm taking a combination of 2 to among all the n, considering all combinations. So I can generate a matrix of sequence similarity. Okay, you can use the cholesterol W or the HS spread matrices. Average branch length, like which data set is much more closest. I can take up the set and find out the average branch length, taking it from here. Okay. So average branch length and the sequence similarity. But if I'm building on the some proteins or some ligands or these two proteins are connected together in a complex, then I can find out what is the energy between the two interacting proteins. Alright, more is lower energy means more stable complex. Okay, they are very native partners, very stable partners. Okay, number of interacting residues, how many residues are always in interaction, types of chemical interactions which are normally used. Okay, mutations in the sequences. If some interactions are going on, now these interactions are because of some mutations existing at those residue positions, or they are actually wild type residues. Okay, so it's an interesting observation if we do it further. So we are extending the discussion of sequence similarity, branch length to protein-protein interactions, where you are considering like energy, number of scoring residues, and what are those residue positions, mutations, and along with the like what kind of bonds are there. Okay, number of sequences. Now, this number of sequences is an important step to consider here. The reason is, the reason is like if a more number of sequences are there which are not at all related also, then the entire part of calculating the observations or considering the mutations, and deciphering the mutations in the sequence data set will go waste. Reason is because I don't have ample data set to consider every single mutation available in the nature. But if my data set is complete, I am having a substantial number of representative sequences from various classes. Then I have enough number to classify the mutations. Now formula used to calculate energy. This formula used to calculate energy is a like important part. But many people what they do, they like keep on correlating the energies even if they are calculated using different formulas, different tools, different strategies. Okay, so if the algorithms are different. How can you put them together at the same par? Okay, so you need to understand that properly. Lack of knowledge about the interconnecting protein molecule. See, normally what happens if the two proteins says this one and this one, it's found to be connected. They are actually involved in interaction with each other, but with some other protein. But with some other protein as a mediator. I cannot say that this protein and this protein are not at all connected, because there is no direct mutual connection. It may be channelized using one or two mediator proteins. Okay, so we need to consider that scenario as well. So whatever the data set you are considering, it is certainly meaningful if you consider all the interacting proteins and their mediator proteins. So only it is the key goal to consider as much protein as possible. Yeah, like in the last lecture we have been discussing why it is important to consider maximum number of proteins which may be possibly like the key partners. Interconnected water molecules are not always usually considered. Sometimes what happens, two proteins are being connected using a water molecule in middle, because this H2O here, there's a hydrogen bond formation between this protein and the other side of the protein, okay, using the other hydrogen, but there's a connection. So these intermittent biological waters, we are calling it as biological waters. These biological waters are crucial to govern the protein network, but we may ignore that as well. Okay, so it is a crucial aspect. All of you think about these questions. What is the main purpose of metabolic pathway reconstruction models in systems biology? To analyze the structure of DNA molecules? Of course not. To analyze, investigate protein interactions. It makes sense. Understand the cellular networks of biochemical reactions within an organism, like organism's metabolism. That's the primary purpose. Option C becomes the right answer. To study the dynamics of cellular membranes. Of course not. So option C becomes the right answer. Which step in metabolic pathway reconstruction involves filling in the missing reactions or enzymes to complete the metabolic network? Which step? Okay. Genome annotation, no. Model assembly, no. Gap filling, yes, of course. So that's the part which is involved in the missing reactions. Remember, we are connecting the like a missing dot somewhere. Like two proteins are being connected with some other protein. So all these missing gaps we need to find out. This is termed as gap filling. Metabolic pathway reconstructions model can always be wrong. Can also be wrong. True or false? Answer is true. If the data considered is not properly correct, or you are like considering some extra proteins, or excluding some crucial proteins which are required, the reconstructed model can even give you wrong answers or wrong results when you check them lastly. Okay, you need to rebuild the data set properly. That's the only strategy which you can use. Okay, so if the answer comes wrong, you need to reconstruct the data set. So see, now we'll be extending our ongoing discussion to the mapping of reaction networks. That is how metabolic or catabolic reactions are guiding the entire pathway, or how if multiple reaction steps are there, how do you connect them further, and how to check the validity altogether. So see, as we have discussed earlier, the metabolic pathway reconstruction strategy includes 6 steps. That is data collection. See, we are collecting all sorts of data as we have discussed. We should not leave even a single one. Reaction identification means what kind of reactions could be there. Stoichiometric matrix reconstruction, the construction aspect. This just represents like the matrix associating different proteins or the metabolites. Okay, and along with the how much they are included in the reaction. Just like balancing the reaction. The balanced reaction means 6CO2, so 6H2O plus gives C6H2O6. So 661. Okay, just like the matrix you can make it as. So how much it is required to drive further? Network visualization. See, you can use the visualization strategy to like, see the network. Why it is important? I'll be showing it soon. Annotation and contextual information. Under what context the reactions goes on, what are the key parameters, what are the kinetic concerns, the kinetic energies and other things, all the parts you put it here. Okay, and the iterative refinement, of course. This iterative refinement makes a significant input to just increase the overall accuracy of the constructing model. Okay, when you go for the simulation, so once you simulate the data set, you understand that the constructed network is not performing the way you want. Means something is wrong there. So you can tune up the things. So see, like illustration aspect, like normally we put metabolic pathway maps, where like metabolites or the proteins or any kind of interacting aspects, interacting the molecules are kept as nodes, and the connection between them are termed as edges, just like a basic graph theory. So those of you who have gone through graph theory as such, like they might be exactly knowing what we are discussing. Okay. Now the graph-based networks is helping us all together. Like whenever we are considering simple metabolic pathway, we don't show the direction in the graphs, which component drives which other reaction. But when we are showing the graph theory networks, we are always showing the representation, like which protein is guiding what other protein. Okay, just to get some sort of directionality in the complete biochemical pathways. But the circular pathways, see the circular pathways are used whenever like a more number of proteins are there or more number of edges are there. Okay, so we arrange all the different components of the pathways as like concentric circles one after the other. Okay, and that way we keep on connecting many proteins also in a small space, even after showing all the given information. Okay, so it becomes easier for us to analyze the complete network. See, what are the metabolic pathways working in our body? One is catabolic pathway and one is anabolic pathway, as you know. So catabolic pathway means something which is breaking up the things. Okay, amylase breaks down complex starches into simpler sugars. Okay, like anabolic pathway is just like building up the bigger molecules from the smaller ones, like amino acids to proteins. Alright. Now see the part, now we are going deeper in the things. A metabolic pathway has several steps. See, just like to start with, I'm showing a part here. Starting molecule, like A to B, B to C, C to D goes on. Reaction 1, I'm calling it as reaction 1. Okay, because this may be a key component. 2, 3, with enzyme 1 and enzyme 2, enzyme 3. When it goes like this, actual details, when it comes some in the research articles, I show reactants. This is the reaction intermediate. See, here I am putting it common intermediate. Reason is here the branching point is there. This gets bifurcated into two. Okay, so this reaction goes on the other side, another reaction goes on the other side. Okay, so this is a common intermediate. Whenever the complete metabolic network goes on a cell, there is a heavy load on the supply of this molecule because it has to maintain both the reactions down to it. Okay, so the downstream pathways, the reaction steps down to this step are termed as downstream pathway. This downstream pathway is heavily dependent on the supply of this molecule or the initial substrates altogether. Okay, making the first reaction steps as the rate limiting steps. Okay. So see, whenever we are analyzing all these parts using different tools, what we do is we take genes, gene products, the proteins, genes, DNA sequences, okay, and then we combine that with the metabolic cycles, metabolic pathway database, pathways, reactions, compounds. We put them all together using different softwares, and this is exactly the same algorithm or same strategy used by different tools. So on the other side, when you are taking what it yields to me, what it gives to me, it gives me pathways, reactions, compounds. Okay, combining the detailed information from the part and connecting the missing things. Okay, was this the strategy used to bridge up the gaps existing in the current literature? Okay, and see, gene product, genes, and genomic path map passage. So only concern is to identify the putative networks which are existing in the complete biochemical network of the cell, but are currently not known to me. Okay, so when I'm clubbing or like putting all the information together on a single table, okay, then I'm able to understand it, decode the missing links. See, now extending the discussion further, like when we are putting up all the like reaction steps, all the pathways together using a software, we can find out the missing links. All right, all it all it helps us to bridge the complete missing gaps to make extend the network properly. Okay. So whenever you are building, we are inferring the complete metabolic reaction steps that can be analyzed. Okay, that can be analyzed. Okay, this is the key word. So infer metabolic steps and the pathways that the genes are involved in. So list down all the biochemical pathways, list down all the genes, and then keep on correlating where the genes can be involved and what are could be the missing genes. To extend it further, find out the reactions, reactants, intermediates, and the products completely. So now when we, just like we analyze the factors which govern the accuracy of protein-protein interaction, understand that whenever you are building a metabolic pathway, here also some key things are there which are crucial factors to determine the overall accuracy. Understand that many genes may not be rightly annotated. They are written something with some function, arginine kinase in the pathways, but they may not be arginine kinase. So it's our primary motive to understand the motives and confirm the motives, validate the gene's function, and then only like, confirm that part to take it further for the analysis. Second part is some genes, some names which are like multiple part is provided. Several names are provided for the same enzyme. Alright. So these several names normally trouble the complete network. Alright. Moreover, like several reactions, like sometimes occurs in the multiple pathways. So these reactions should be properly taken care of. Okay, well these reactions are the core backbones of several different biochemical pathways. Okay, so whenever we are putting some gene somewhere, we need to always remember that we need not ascribe the gene for the same function. We need to consider multiple functions in picture. Moreover, the major trouble is like alternative pathways also exist in various different species. These are termed as salvage pathways. Okay, besides the normal pathway, salvage pathways also exist. Okay, so when we are taking all of them consideration, like we need to get an overall picture of the entire metabolic network. Okay, which is an important part. Like see a small step here. Whenever I'm considering like reaction 1 to reaction 6 here, like T1, pathway 1, can also break down a pathway 2 and pathway 3. Now understand that if a pathway 2 is present somewhere, pathway 3 is present somewhere in the same species. And I am able to build up a mechanism, build up the part whenever I am analyzing my data. I am understanding that pathway 3 is connected to pathway 2 using R3 and R4 connected here. So I am able to build a super pathway. This is termed as a super pathway P1, which is connecting two sub-networks P3 and P2. Like that, the analysis is done. We are always connecting the small pathways. So now let us understand the cell check MCQ and give the answers. Which of the following techniques is commonly used for visualizing and representing a reaction network? So genome sequencing is of course not the right answer. Flux reconnaissance FBA is also not the right answer. Circular layout makes sense. So option C is the right answer. Which of the following metabolic network construction steps yield the accurate results on the basis of detailed available biological data? Remember that we always need to consider the detailed available biological data. Yield accurate results. Collecting relevant data, identifying individual reactions, performing statistical analysis. So whenever we are doing a statistical analysis at the end, just like we discussed the p-values, okay, that is allowing us to consider the most like valid like reasoning or the most valid network solutions which can be constructed from a given data set. Multiple solutions of the graph may turn in, but statistical results help us to identify the accurate results. So option C is the right answer here. Conceptual questions. This just like I discussed in the last part also, metabolic reconstruction, metabolic pathway reconstruction models can be wrong also if wrong data set is considered or any of the step is wrongly taken up further. Because all this accuracy is taken up from 6 individual sub-steps. Anywhere if you're doing something wrong, the inaccuracy keep on moving. Okay, so inaccuracy is propagated further to the next step. So, and the most important of this is the wrong data set. If you take any gene extra or you discard some like important genes, we are like decreasing the overall accuracy of the metabolic network. The last, so in the last phase of this session, we'll be analyzing how Biocyc and the KEGG database or the individual their modules have been used for the metabolic pathway construction, especially how we can deploy their modules to build the gaps. Okay. Now remember that KEGG is just including metabolic pathways along with the associated genes and compounds. Like, so whenever you need some information on certain missing bridges, missing connections, we normally take the help of KEGG modules. Metacyc is further curated database of experimentally determined metabolic pathways and enzymes. And this Metacyc is a part of Biocyc pathway database. Biocyc is a collection of several different databases. Okay. These databases are constantly expanded and updated. Expanded means bridging up the missing links to research papers and then constantly updating the databases. And these databases allow for the analysis and visualization of metabolic pathways. Alright. Whenever you are finding the key metabolites and enzymes, we need to update the database and we can understand the flow of metabolites through a pathway. So all this information put together gives us the overall insight into the like function and regulation of a metabolic network. See, when we start discussing about Ecocyc, It is only meant for E. coli, as you know from the earlier sessions. And now it is used for microbial genome annotations, that is finding the promoters. Okay, study like coordination of E. coli metabolic enzymes into structural protein families. So it gives the links to the other different databases, finding operons, finding promoters, finding protein functions and interactions, or predicting the protein-protein interactions and protein fusion events. It also gives the references where it has been reported earlier. Okay, to analyze the connectivity relationships of the metabolic network, how it could be there. Enzyme activation and inhibitions, repetition and multiplicity of like elements such as enzymes, reactions and substrates. Okay, so this is how the complete ecocycle looks like. See, so I can search several different things here. See, it's all a thousand of different databases connected here. Now, see if I search the summary for this Biotrophia defectia, I'm searching it here. See, human blood, post Homo sapiens, human microbiome, body weight is present is blood. See, total genes, different things which are involved in undisplayed quantico replicons. It is from the minute level, it is taking it all together. So in the NCBI part, we need not search the genome where it is exactly encoded. We can directly search in the Ecocyc. Okay, if it is some information is there, right? So more detailed table is listed here. See, pathways, enzymatic reactions, transport reactions, polypeptide, protein complexes, enzymes, transporters, compounds, transcription units, RNA protein features, and gene ontology terms. So many details given all together. Okay, at one shot. That's the entire advantage of all these databases. All these databases are meant to connect several different databases. As we discussed the unifying theory, people are unifying various databases or stitching them all together to get the meaningful information. Alright. Enzyme interactions, key molecules which are crucial for all of us, either the activators or inhibitors, cofactors, or alternative substrates. Okay, that the enzyme will accept. Because the enzyme active site is, like we are calling it as promiscuous. Okay, if this substrate is not there, the enzyme will work with other substrates as well. So the active site is promiscuous, interacting to various substrates. It is termed as promiscuous active site of an enzyme that binds to many substrates in such multifunctional enzymes. This is a crucial part. So only that enzyme gets repeated for multiple functions as well, as you have been discussing in this session. This is a Metacyc database. See, non-attendant metabolic pathway database. See? It includes the experimentally determined metabolic pathway. See, all reactions, alternative substrates, cofactors, prosthetic groups, activators and inhibitors, like subunit structure, molecular weight, PI research data, literature citations, species. Everything is listed together and connecting so many pathways. See, so it's just a step further. Metabolic regulations, transcription, regulation of gene expression, everything is gone. Now see, for example, how the pentose phosphate pathway looks like. Building it up through various different metabolic pathways connected here. When I am putting up all these reaction networks, okay, see the substrates put together, okay, and the edges are showing the reactions. All these reactions connected together using a like arrows, which is providing a directionality to the complete pathway. The plus and minus here, indicating the activation and inhibition. See? See how the regulation of pentose phosphate pathway in more detail, connecting more enzymes. Now tell me the answer. See, if you observe the reactions very carefully, you will find that the number of genes is smaller than the number of biological reactions because multiple genes are involved in several different pathways, several different reaction steps. Okay. Enzymes are still not found for some of the reactions and the research is still going on. This means connecting the bridges, connecting the known pathways, okay, finding up the missing gaps. And the second one is the major trouble for all the metabolic engineering experiments. People are working out to know the enzymes or decode the enzymes for which proteins are present, but still we don't know the function. Where exactly to connect it as? This is the part of KEGG. The KEGG symbol as Kyoto Encyclopedia of Genes and Genomes, the Japanese server. It integrates current knowledge of molecular interaction networks, information about genes and proteins, information about chemical compounds and the reactions. So, see what all it integrates now genomes, genes, and pathways of completely sequenced organisms. Okay, because otherwise it has to do annotations and the manual annotations. So the functional annotation for each gene by comparative genomics. Okay, as we like discussed a few sessions back, it has to keep on comparing the various different genes in different species, like homologs or orthologs, and the gene similarity transfers. Okay, pathway connection, reconstruction based on the annotation. So trying to find out the missing gaps. A system for computing and comparing biological networks from molecular interaction data. See, we can work out all of these. It is an extended server having numerous modules. This is how the complete, just in a nutshell, how the KEGG database work out. The expression data, genomes and the genes, all the pathways are there. Putting all this expression and the genomes and genes all together in a single table for the connected pathways and linking up the chemicals and the reactions using the orthologs and the sequence similarity part, extending each and every part everywhere and that too with logical networks. Okay, so if I ask you using KEGG, tell me the enzymes which are involved in the glycolysis of Homo sapiens. Homo sapiens are the humans, okay? See, I can go to the KEGG database, the KEGG pathway database here, see? I can go to KEGG table of contents modules which are available in KEGG, go to KEGG pathway databases and see, I can immediately turn it on. They click here on the glycolysis. See? Then the next part is suppose if I want to know generate possible pathways between two compounds, which the common like people do. Like if I have the substrate and the product, I don't know what all the reaction steps are going on in between them, or what are multiple possible pathways through which this substrate can be converted to this product. I can work out, put the substrate here and the product here. See, okay, and see. Enter cutoff length, okay, no relaxations and other things. This is just to if I want to be strenuously doing it, with the completely this part, or can I allow some liberty somewhere? The threshold, see, show me the result. So changing these parameters will show change the results as well. See? Let us understand how much we have understood so far. What are the benefits of using Ecocyc pathway prediction? It is a comprehensive database of metabolic pathways for E. coli. It can predict metabolic pathways of other organisms by comparing their genomes to the Ecocyc database. Both the statements are correct. Because we know that we are checking the other genes also of other species to check how much they are similar to the ecosaic genes, E. coli genes, and get some meaningful information to connect the missing links. It's a valuable tool for metabolic engineering and for studying the metabolism of organisms. This is also correct. Answer lies all of the above. What are the benefits of using Metacyc pathway prediction? Accuracy of the predictions depend on the quality of the organism's genomes. Okay, that's of course meaningful statement. The predictions are not always complete, and they may include all the active enzymes of the organism. Yes, that is 100% there. Like all the active enzymes or the pathways may not be included in the pathway. Okay, so the predictions are not always complete. Some missing links will be there. You need to trace them. These predictions are not always accurate and they may need to be adjusted based on the experimental data. If you studied this session properly, you might have understood that this we are always validating the experimental data with the constructed metabolic network. We understand that whenever we are doing the experimental part, we find that the values which you are getting at the end of the pathway, okay, in terms of the yield of the enzymes or how the complete stoichiometry of the network will look like, we do not get the same things as we are computing. So some other genes or the metabolites or the enzymes may be involved. Okay, the answer lies all of the above. So now tell me the question: Ecocyc and Metacyc are manually curated databases for biological data mining. It is of course true statements. All the errors, all the missing links, all the inaccurate definitions regarding the functional nature or nutrition of various enzymes. Okay, it's always corrected by like manual curations. We manually correct the things. Okay, is it is the results reliable? The result reliable or does it need to be experimentally validated? Of course, there is a need to do experimental validation. The reason is whatever we are getting the answers here, there is always a need to proofread the things that we are getting. Thank you. I hope you might have enjoyed the session here. Okay, and I'll look forward to for your continued viewing of this channel. I hope you will pass it on to your like interested friends as well. Thank you.